On behalf of Cohen Veterans Bioscience, it's my pleasure to welcome you to the Cohen Veterans Bioscience webinar series. This is a monthly program sponsored by Cohen Veterans Bioscience, the brain research and biology communities. Cohen Veterans Bioscience is a nonprofit research alliance whose mission is to transform research in veterans' mental health through a translational research approach. We are focused on understanding the molecular underpinning of diseases, such as PTSD and TBI, to develop better treatments and diagnostics. Before I introduce the statistics, this program is being recorded and will be archived in the Cohen Veterans Bioscience YouTube channel and website. The presentation will be about 40 minutes in length to allow for a Q&A afterwards, so please use the Q&A button to post a question at any point during the presentation. Today our speaker is Dr. Israel Liberson. Dr. Liberson is a professor of psychiatry, psychology, and neuroscience, and the director of the Psychiatric Residency Research Track in the Department of Psychiatry at the University of Michigan. Dr. Liberson's primary research interests centers on emotion, stress, and stress-related disorders such as PTSD. In 1992, he established a PTSD program at the University of Michigan in Ann Arbor VA Medical Center. He also co-founded the Trauma, Stress, and Anxiety Research Group at the University of Michigan. Dr. Liberson serves on the NIH and VA study sections and served as a reviewer of medicine in the Department of Defense congressional reports, as well as various international funding agencies. In this webinar on processing PTSD, we focus on potential abnormalities in the hippocampal-prefrontal-thalamic circuits of PTSD patients in animal models. It is my honor to introduce Dr. Israel Liberson.
Good afternoon. Thank you very much for the kind introduction. Today's webinar, we're going to focus on contextual processing in post-traumatic stress disorder. The focus of the webinar will be not on epidemiology or the treatment of post-traumatic stress disorder, but rather on understanding of the underlying neural circuits that could be involved in the development of post-traumatic psychopathology. Now that obviously requires some kind of general knowledge of the disorder, and I just briefly touch on its symptomatology, as well as on the background biological studies that have been done over the many years, decades as a matter of fact, in PTSD. However, the bulk of the talk will be focusing on what is happening in the brain of PTSD patients. What could be the mechanism involved? What are the neural circuits that underlie development of post-traumatic stress symptoms? And naturally, identifying those circuits and understanding those mechanisms is the first critical step to developing informed, neurobiologically informed treatment strategies as well as secondary prevention. So let's start with, first of all, reviewing very briefly PTSD symptoms. Post-traumatic stress disorder, it's a common disorder, occurs in approximately 7% of the population, more common among women than in men, and it's characterized by 4 sets of symptoms. This is intrusive re-experiencing symptoms, which involve intrusive memories, nightmares, intrusive recollections, and then the behaviors that patient exhibits in order to prevent themselves from experiencing these intrusive symptoms. And that's the avoidance of any reminders of the trauma or any situation or places where these reminders can occur. The third set of symptoms is negative alteration in cognition and mood. This is a relatively recent addition to the diagnostic criteria. Before that, these symptoms were basically combined with avoidance symptoms. So the symptoms is not new. The way that we're thinking about it a little bit new for DSM-5. But it basically highlights the fact that there is a set of symptoms like emotional numbing that are probably not similar and maybe don't come from the same source as the avoidant behavior. And these symptoms today we describe as negative alteration in cognition and mood, such as inability to experience normal responses, very negative overall attitudes toward and perception of the world. And finally, the alteration in arousal symptoms and hyperreactivity, so it's hyperarousal, hypervigilance, sleep difficulties, and so on. Additional symptoms often seen in PTSD is what we would say reckless behavior or re-traumatization that almost paradoxically occur in some PTSD sufferers where people get re-exposed to trauma, and which was very difficult to understand within the more traditional conceptualization of this disorder. Now, I'm referring to these sets of symptoms because throughout the talk, as we go through various models and neurocircuits involved, we will try to trace what are those models that involve abnormal circuitry can explain among the symptoms, what it cannot explain yet, and how that propelled the research to continue search for novel explanations and identify novel targets. PTSD had been studied extensively over the years. And the most common systems that have been identified as abnormally functioning in PTSD, these are peripheral systems that are associated with either reactivity to stimuli, such as psychophysiological findings of increased baseline heart rate or increased blood pressure, or enhanced heart rate response or skin conductance response or EMG response to trauma-related stimuli, exaggerated acoustic startle, and more. These psychophysiological symptoms basically suggest that PTSD patients are at least peripherally hyperresponsive to trauma-related stimuli. In addition, sleep abnormalities have been detected, and while there is quite an extensive literature on sleep, the evidence is still not conclusive, with probably majority of the evidence preponderant suggesting that there is an overall disrupted sleep and less efficient sleep, included shorter, more interrupted REM periods, and so on. Furthermore, within the peripheral systems, the plasma levels of peripheral hormones associated with threat response have been measured. And interestingly, the two major sets of findings emerged. One is increased catecholaminergic responses, enhanced catecholamines levels in response to traumatic stimuli, but also overall increased 24 hours urinary epinephrine and norepinephrine secretion, as recently changes in potentially in adrenal receptor beta 2 genetic predispositions, potentially changes in alpha-2 receptors in platelet, and so on. Together, it had been suggested that together psychophysiological finding and catecholaminergic finding that PTSD represent a kind of hyperadrenergic state. And we will return to this point throughout the talk, because this is probably the most commonly replicated finding, and whatever neurobiological model of circuits we're going to be trying to examine, at some point it has to accommodate and to integrate those peripheral findings into one single picture. The other component of peripheral hormonal responses to stress and threat is hypothalamo-pituitary-adrenal axis. And here, mostly unexpected and somewhat surprising findings have been reported in PTSD. These involved increased glucocorticoid receptor sensitivity, as expressed by enhanced dexamethasone suppression test, as well as potentially increased glucocorticoid receptor in the lymphocyte, low 24 hours cortisol secretion in the urine maybe, as well as genetic findings of changes in FKBP5, which is chaperone for the glucocorticoid receptor gene. So the picture in HP axis is quite unique and distinct from other disorders such as depression, whereby rather than having low glucocorticoid sensitivity and high cortisol level that we see in depression, in PTSD, what is commonly reported is increased glucocorticoid receptor sensitivity and potentially even low urinary free cortisol secretion. So this particular profile is also critically important for us to keep in mind while trying to integrate brain circuitry and try to explain how these signals of abnormal functioning in the brain can contribute to, on one hand, hyperadrenergic state in PTSD that I just mentioned before, sleep abnormalities, but at the same time, abnormal glucocorticoid responses and HP axis characteristics. Now, there's another set of evidence that's starting emerging, such as deficits in potentially neurosteroid function, endocannabinoids, NPY, and DHEA, but these are initial findings, and they need to be further replicated to be considered as characteristic of PTSD. So after this general introduction of PTSD as a clinical disorder with subset of peripheral abnormalities, we will go and review a little bit with historical perspective what is considered or what had been the various models of PTSD pathophysiology in the brain, because that's the critical point that needs to be explained. The PTSD symptoms cannot be generated by hyperadrenergic states alone or by HP axis changes, whether these affect the brain or the final outcome is of the brain. But what drives the behavior and abnormal symptomatology is some type of changes in the brain circuitry that produce altered function, and this is the core of the disorder that needs to be explained. The talk today will focus around 4 circuits that have been identified, and from right lower corner, fear learning circuits that are specifically focused on the amygdala. We're probably the first one to identify as potentially implicated in PTSD, and I'll get to that in a minute. Through the threat salience detection circuitry, which involved amygdala, anterior cingulate, and anterior insular operculum, that are responsible for detection of salience in general and threat in particular. Then talk about emotional regulation and executive function circuitry, which involves dorsolateral and dorsomedial prefrontal cortex and ventrolateral prefrontal cortex that's involved in regulation of emotional responses. And finally, converge on the more recent arrival to the scene is a contextual processing circuit that, while by itself is not recent, had been studied for years. Our conceptualization of contextual processes as key deficits in function in PTSD had been real recent, and in a way in response to some of the deficits of other explanatory models. As I mentioned before, the studies of central processing of what's happening in the brain in PTSD started with the development of tools that we call functional imaging that can perform studies in PTSD patients in vivo. And these started in early 90s with a set of studies, and I put a couple of them here on the slide. On the upper right corners from Scott Rauch's and Lisa Shin at that time, study in using positron emission tomography. Study under that is from our group using single photon computerized tomography, basically demonstrating the blood flow in the regions of amygdala specifically in PTSD patients are abnormal as when it's compared to control in a Scott's and Lisa study, or to combat control and trauma-exposed control and non-trauma-exposed control in our study when those patients are exposed to the trauma-related stimuli. So this was the first set of studies in mid-90s that have been implicated in PTSD pathophysiology. That have implicated amygdala specifically in PTSD as brain region that have clear, distinct, different function. Now, the other set of developments within the field of neuroscience in general and affective neuroscience in particular is that approximately the same time the animal studies of emotional responses came to fruition. And that happened in response to recognition that one can model a basic emotional responses such as response to fear or threat inducing stimuli in animals, and that will allow us to study the basic anatomy or neuroanatomy of these responses. So the animal studies have been implicating, of course, amygdala in emotional response to fear. But beyond that, animal model at that time developed by Ledoux, McGaugh, and others have suggested that amygdala is a critical component of fear learning mechanisms. Fear learning whereby animal learn what to fear and remembers what to fear, what specific cue to fear. Now that provided an excellent explanatory model to try to link the exposure to trauma and then memory of the trauma to neurobiological processes. So if fear learning is abnormal in PTSD, that suggests that PTSD patient might learn to fear particular stimuli more than somebody else. And if so, then one should look into the amygdala of PTSD patient and find abnormalities in this process. In the lower left corner of the slide, you can see the various models, or I'm sorry, the graphic depiction of amygdala nuclei and projection between them. And a lot of work has been done by multiple investigators delineating those pathways and identifying a specific pathway within amygdala or basolateral complex of amygdala that participate in learning of fear. Now, on the other hand, if you look on the right side of the slide, it demonstrates two alternative possibilities. So the graphic depiction here of what we call conditioning and extinction of fear, while the red line demonstrates the memory of the conditioned cue or condition trace, and the green line demonstrates learning of extinction means learning that the same cue in different circumstances does not necessarily represent fear. Actually, is usually will not be associated with fear signal. So one can see that if white line on the top represent behavior, it can be a mathematical or arithmetic combination of memory of fear as well as a memory of extinction or safety associated with the stimuli. So in the normal circumstances, when we learn the fear, the behavior expresses this of fear expression develops. But then when we learn safety, the behavior normalizes. This model suggests that either if one learns fear in an abnormally high or exaggerated way, or fails to learn safety appropriately, in either of those conditions, the strength of the memory trace of fear and safety will not be the same, and the organism will exhibit exaggerated fear behavior. This was a very interesting and accessible model that could be studied in animal, and it was almost immediately adopted by the scientists that studied basic mechanism of PTSD, and actually it contributed to a lot of very, very important discoveries on the function of fear learning and safety learning. These were primarily concerned with signaling within amygdala and basolateral complexes, as I mentioned before. Interestingly, as you can see on this next slide, while this model had been applied to PTSD and kind of widely accepted in the study of neurobiologists, by neurobiologists, when you applied it clinically, the clinical studies were equivocal at best. So there's at least the same amount of studies that have failed to demonstrate exaggerated fear responses in PTSD as those who did demonstrate those responses. Further on, one could argue that maybe the PTSD fear learning mechanism is not abnormal in PTSD. But fear mechanism is normal, but the trauma was so exceedingly stressful or threatening that the experience was hyper-conditioned because of the excessively arousing and threatening experience of trauma. However, if one looks epidemiologically in prospective studies, one sees that prospective studies, and I'm talking about studies that recruited subjects in the emergency room following exposure trauma and followed them up to see how many of them develop PTSD. These studies actually reveal that severity of this trauma, as measured objectively in the emergency room, does not necessarily predict the development of PTSD. So failure to, on one hand, to demonstrate this hyperconditionability or failure to, or diminished extinction in PTSD patients on one hand, and then this evidence that the initial severity of the trauma does not necessarily predict PTSD, raised first set of serious questions about fear learning mechanisms as explaining PTSD pathophysiology. At the same time, as functional neuroimaging studies continued to develop, there had been reports start coming in demonstrating that amygdala hyperactivity is actually not specific for PTSD, but can be seen in general fear in patients with specific phobia, and so on. So together, that's also raised the question about uniqueness of this amygdala hyperactivity that we have identified PTSD, and raised the possibility that what we see in PTSD is expression of intense fear in the amygdala, but not necessarily the source of the abnormalities that generate this exaggerated fear. Furthermore, if one thinks conceptually about fear learning abnormalities and the model, if indeed the deficits are in the fear learning only, that could explain exaggerated psychophysiological responses I mentioned before, and exaggerated emotional response to trauma-related stimuli. It could explain intrusive symptoms if the memories are extensive and indelible and difficult to extinguish. It can explain even the avoidance symptoms, such as avoidance of behavior, which is secondary to those extensive and exaggerated intrusive symptoms. But still, it cannot explain really the hypervigilance of PTSD patient. Means going into the place where there is no specific cue and looking for the cues of danger. It definitely cannot explain emotional numbing. It cannot explain intrusion which are not triggered by trauma cues, and definitely not re-exposure to traumatic or dangerous situation, that if fear learning was exaggerated, the PTSD patient should have been more or less likely, if anything, to get exposed to dangerous situations. Now, to be fair to this model, the amygdala findings were not the only one that have been reported. And as I will show in a minute, there was also findings in the ventromedial prefrontal cortex, and specifically diminished function ventromedial prefrontal cortex, that have been reported in PTSD patients. And investigators tried to incorporate these findings together with hyperactive amygdala in argument that these findings demonstrate failure to retain or recall extinction memories. However, if that's the mechanism, this mechanism is quite different from the mechanism of fear learning, and requires a little bit different understanding of what is the pathophysiological process and what exactly the function of this memory of fear learning or fear extinction, and how is it different from the memory of the fear trace itself, and so on. And I'll get back to it in a couple of minutes. As I mentioned before, this concept that fear is, as a matter of fact, that fear learning is extremely important mechanism that allows us to avoid dangerous situations, you can see on the top of this slide. But it also requires to be well regulated and not exaggerated, as you can see in the upper right corner. So the balance between learning fear response to avoid dangerous situation and regulating fear response had been also considered in neuroimaging literature. I would say neuroscience literature. Within this construct, the role of the medial prefrontal cortex became even more important, because there have been studies that were coming from the basic neuroscience that clearly delineated the role of medial prefrontal cortex in regulating amygdala responses. The studies below are from Gregory Quirk and on the left, and those from Anthony Grace, electrophysiological studies as well as behavioral studies that demonstrated the projection from medial prefrontal cortex to amygdala actually inhibit amygdala responses. This occurs because the projections, although they exit to the projections, they end up in intercalated neurons within amygdala that are GABAergic, that in turn inhibit the output of the amygdala. This became a very interesting model that suggests that maybe this particular circuitry of diminished emotional regulation is responsible for some of the PTSD pathophysiology. Now, at the same time, as basic science work in rodent models was implicating medial prefrontal cortex, or specifically ventromedial prefrontal cortex, in regulation of amygdala. The more cognitive-based studies in human clearly demonstrated that emotional regulation in humans involves both medial prefrontal cortex, which you can see on the right upper corner, but also dorsolateral prefrontal cortex and ventrolateral prefrontal cortex. These are the studies that performed by Gross and Ochsner and others, which have directly asked individuals to try to regulate their emotions using various skills such as reappraisal or suppression and so on. These studies implicated all of those structures, such as MPFC, DLPFC, and VLPFC, in down-regulating amygdala and emotional responses. So together, these evidence gave a raise of alternative hypotheses, basically implicated alternative brain circuits in PTSD. This is a hypothesis of diminished emotional regulation or diminished executive function. The hypothesis here suggests that if there is a deficit in the function of medial prefrontal cortex, DLPFC or VLPFC, they will not be able to regulate amygdala signaling when it comes up, and that will result in potential number of symptoms that we see in PTSD. For example, memory deficit about trauma, exaggerated emotional responses to salient cue could stem from the failure to regulate emotion rather than from abnormal learning of this emotion, as well as irritability and impulsivity in general, because those regions are supposed to regulate overall emotional responses. Now, interestingly, some of the same regions that are implicated in emotional regulation, these are regions also implicated in executive function in general. Since executive function, it's a general set of functions, such as avoidance of danger and resisting of distraction, ability to keep information in mind, switch between tasks, planning, and so on. So the converging evidence suggests that there is a common set of prefrontal regions that are implicated in the regulation of both affective, such as emotional regulation responses, but also non-affective functions that are fall under the executive function category. If that's true, one would say that one should look for deficits within those networks in post-traumatic stress disorder, in addition to the evidence of diminished signal in ventromedial prefrontal cortex. And indeed, as you can see in the lower right corner of the slide, there is initial evidence from McNamara and others that PTSD patient might be deficient in recruiting, at least before treatment, those lateral prefrontal cortex regions when asked to reappraise negative stimuli, the deficit which is eliminated following effective treatment. There are other studies that use functional connectivity as well as other network efficiency and so on, techniques that also demonstrate the potentially deficits in attention network involving those lateral prefrontal cortex, ventrolateral prefrontal cortex, and so on. However, once again, on the basic neuroanatomical level, it is difficult at least to describe the circuitry of emotional regulation as coherent single neurocircuitry with various components of it, such as VMPFC, DLPFC, and VLPFC, all function in concert. And on the cellular level, there is really not enough information about what defines this neurocircuitry. Conceptually, diminished emotional regulation can explain some PTSD symptoms, such as exaggerated emotional responses to trauma stimuli, exaggerated psychophysiological reactivity, but it really cannot address the intrusive symptoms of PTSD. That cannot explain hypervigilance, emotional numbing, or re-exposure to trauma. So how does one understand this hypervigilance that I had mentioned before? Hypervigilance is tendency to look for a signal or threat signal in any environment, even in the absence of the cues. This type of process suggests that there is some kind of a threat detection that is abnormal in PTSD, not necessarily in presence of any cues. The system in the brain that have been linked to threat detection tightly, or as tight as possible, is what we would call Salience Network. Now, this type of intrinsic connectivity network studies have been developed relatively recently and have demonstrated that brain regions are functionally connected at rest and during task. And those regions that functionally connected can be divided into a number of networks. One of those networks involves insula, or anterior insular operculum, dorsal anterior cingulate, and amygdala. And this network is coined Salience Network. But what it does, it detects salience in the environment, whether threat or reward, and it also focuses attention and recruits homeostatic systems to respond to them. As well as some would argue that it also triggers automatic motor routines in response to those salient stimuli. This network actually involves some of the original culprit of amygdala that we have mentioned before, but also implicates regions as dorsal anterior cingulate and insula, and explains potentially the source of hypervigilance on one hand, or search for threat detection on one hand, and hyperreactivity to threat and fear-producing stimuli on the other hand in PTSD patients. So it was suggested that hypersensitive salience or hyperreactivity in the node of this network, or potentially enhanced connectivity between them, can lead to this hypervigilance and exaggerated physiological reactivity and disproportionate fear in PTSD. Indeed, there had been evidence. You can see on the left side the evidence from Mary Stein and Martin Paulus at that time, of enhanced anticipatory signal in the insulin PTSD patients. And then in the middle and the upper right side from Lisa Sheen and then from Wolf and Heringa, evidence of enhanced dorsal anterior cingulate cortex in PTSD patients. These evidence suggests that and I have already discussed and showed before that amygdala is potentially hypersensitive in PTSD, that threat detection network could be hypersensitive PTSD as well. Our studies on the bottom have demonstrated their potentially hyperconnectivity between amygdala and insula in PTSD patients. However, while exaggerated threat detection signal could explain hypervigilant and exaggerated physiological reactivity and avoidant behavior in PTSD, we still have to explain intrusive symptoms, exaggerated emotional reactivity, and emotional numbing, as well as re-exposure to trauma. So one can summarize that together, fear learning, emotional regulation, and threat detection circuits can explain many of PTSD symptoms, but they will require to postulate three different independent mechanisms. And they don't share common pathophysiological process, nor do they accommodate pre-existing biological finding, still leaving some of the critical symptoms of PTSD, such as emotional numbing and re-traumatization, unexplained. That led us to think and to consider other potential candidate processes that might be involved. And in the past 7 to 8 years, we have proposed that a different circuit that is regulating multiple functions that we have discussed so far might be abnormal in PTSD. And this circuit involved in contextual processing. It consists of primarily hippocampus and medial prefrontal cortex, but it also involves thalamus, which links in many ways between two of these regions, as well as a regulatory inputs from locus ceruleus. In general, contextual processing is a way for the brain to disambiguate stimuli. For example, if one sees a lion in the zoo, one is very relaxed. And one sees the same lion in the backyard, that's pretty scary. Even if the zoo is built in a way that visually we don't see the barrier between us and the lion, our brain sufficiently distinguishes between two, although the visual input could be very similar. Our brain does it because our brain perceives the context. It knows where we are. So it combines many different elements that assemble into one representation. It says, oh, we're at the home or we're in the zoo. In the zoo, it's safe. In the home, seeing lion is very unsafe. That allows an individual to flexibly respond to stimuli that in one situation can be extremely threatening and another situation can be actual fun, like a lion in the zoo. This particular mechanism also modulates fear conditioning or fear learning and fear extinction, the safety learning, as well as it modulating other critical functions as emotional regulation in humans. If indeed the contextual processing is diminished in some way, it has reduced capacity to modulate fear in PTSD patients, that suggests that PTSD patient may be particularly sensitive to specific cues, specific events, specific context that potentially could explain all these abnormalities, and I'll get to it in a second. So how would we study that? So we used the functional neuroimaging tools to try to demonstrate that using same fear conditioning extinction paradigm. So we exposed PTSD patients and controls to various stimuli. You can see on the top here, we condition them to one of the lights, either pink or blue light. In this case, it's a pink light, but between subject it was changed. And this is a CS plus and CS minus. And then in the second day, we extinguish that. Basically expose them to the same light without shock. And we demonstrated, first of all, on the lower left corner, that in all the patients, conditioning indeed activated amygdala, insula, and other regions associated with fear. And then we demonstrated that after extinction, those activations were eliminated, and there was no difference between PTSD and combat controls. However, when we re-expose them to fear renewal and extinction recall, what happened is that PTSD patient did not recall that as well, both vis-a-vis skin conductance and vis-a-vis their brain activities, their extinction. So they remembered fear more than extinction. And you can see it on the left with, I'm sorry, on the right of the slide with exaggerated amygdala responses, but diminished medial prefrontal cortex responses. So that's not very different from what had been demonstrated in the past. However, we also postulated that if contextual processing is abnormal, rather than fear learning, or even retention of fear is abnormal, during the process, what we call fear renewal, where context is supposed to enhance fear. I'm going back to demonstrate that now we exposing to the same stimulus, but now in the same context as they were conditioned in. If the contextual modulation is abnormal, the PTSD patient has to fail this test as well. The PTSD patient has to have inadequate reactivation of the fear in these cases. If the context is abnormal, they should not be able to recall extinction context or fear context. And indeed, this is what we demonstrated here. This is demonstration that after exposure to extinguished stimuli, combat controls in the fear context had a more significant or larger activation in the amygdala and the insula as compared to PTSD. We further on continued the studies with another way to test context processing is what's called fear reinstatement, whereby rather than using context, we did conditioning and extinction, and do reinstatement using either the same type of electric shock or trauma-related stimuli. And we demonstrate that a response to extinguished cue in PTSD patients was higher, and skin conductance responses were higher following the reinstatement with a trauma cue. Again, suggesting that context invoked by post-trauma cue mediated this enhanced recollection or reinstatement of fear to the novel object. When one thinks about that, interestingly, there is plenty of evidence historically that implicates hippocampal signaling in abnormal hippocampal findings in PTSD. This is an older study that demonstrates that hippocampal size is potentially smaller in PTSD patients. And if you look on our other studies that we have reported, we have demonstrated the connectivity, again, using the intrinsic connectivity network, that connectivity within the prefrontal cortex and hippocampus is diminished in PTSD. If this is true, one can explain that intrusive symptoms and hypervigilance triggered by contextual can be triggered by contextual cue, as well as a traumatic re-experience. Means that people will perceive the re-experience as if it occurs today. rather than in the past. Contextual deficit can also explain emotional numbing. It follows that if somebody experiences joyful event, but the brain perceives the context of the event as sad, context of loss or trauma, they cannot experience the same event with the same degree of happiness. And that's what they call failure to experience normal emotions, or emotional numbing. Finally, it can explain even what we call paradoxically re-traumatization, because failure to understand and perceive the context appropriately doesn't only introduce threat context where it's not supposed to, but it also fails to detect threat context when it's supposed to. Therefore, the PTSD patients might not be able always recognize contextual cues threatening if they're not associated with trauma, and get themselves again into the potentially traumatic situations. In the last couple of minutes of the talk, I just wanted to show you that this contextual processing can be actually studied on a molecular level as well. As I mentioned, it was all developed based on seminal studies of Marin and Cochrane and others that delineated this connection between medial prefrontal cortex and hippocampals as a key regions involved in contextual processing. Now, in the last 15 years, we developed animal model PTSD, trying first of all to model what we call enhanced negative feedback. I mentioned to you at the beginning of the talk that this is a critical component of peripheral finding that needs to be integrated that suggests that glucocorticoid signaling might be abnormal. To do so, we created a model where animals are exposed to restraint, cold swim, and then ether anesthesia, and then left unexposed. And this model has produced uniquely abnormal enhanced negative feedback of HP axis. This is not an animal model talk, therefore I will not go into details how exactly that happened. But suffice to say that as this model was developed, we were specifically looking for enhanced negative feedback and potentially, if we can demonstrate exaggerated acoustic startle, and by that to have animal model that have construct validity for PTSD. Surprisingly, we also found at that time and reported changes in hippocampus, but at that point we still did not know about the contextual processing deficits. And we were wondering, what are those changes in hippocampus that we find, and what is the role of that? However, as we studied PTSD and zeroed more on contextual processing in PTSD, we took the animals from this single prolonged stress studies and exposed them to fear conditioning, fear extinction, and extinction renewal. And lo and behold, we demonstrated the fear renewal in those patients, those animals were abnormal. We then proceeded with the next study where we actually used 3 different contexts and demonstrated that one normal animal benefit from knowing the context of extinction, and this is this lower line here with open circles, the fact that they have been extinguished in a particular context before prevents them from responding to this cue again. The animals who have not been exposed to the same context, they were exposed to all 3 contexts during fear conditioning, fear extinction, and extinction recall. These are the animals in with the squares here, open squares. They show exaggerated fear response compared to the open circles, which show normal fear response. And PTSD animals, whether they were exposed to the same context or different contexts, cannot benefit from the knowledge of the previous context, and all show exaggerated responses. We looked into the molecular components of this model and demonstrated that this particular deficit was associated, or the behavioral deficits were associated with decreased glutamatergic signaling in medial prefrontal cortex and enhanced glucocorticoid receptors in hippocampus and prefrontal cortex, linking together those abnormalities in this general circuit of contextual processing to these two specific systems. Further on, we continued to explore and demonstrated that there is abnormalities in noradrenergic system in those animals. And noradrenergic system in the brain starts from, as many of you know, from locus ceruleus, which contains 50% of noradrenergic neurons projecting to the cortex and subcortical regions. And we demonstrated that SPS animals, the same model animals, also have exaggerated locus ceruleus reactivity as well as abnormal levels of tyrosine hydroxylase in locus ceruleus. I will skip the following slide that simply demonstrate that thalamus plays an important role and locus ceruleus input in the thalamus in regulating the communication between medial prefrontal cortex and hippocampus. And specifically, the critical period of that could be period of sleep, because during the sleep, the updating of the information between the two systems occurs. To summarize what we heard today, we have demonstrated that after exploring potential models of fear learning, threat detection, and emotional regulation, we have developed a novel model of contextual processing modulation deficits in PTSD and demonstrated that contextual processing is altered, and it can be seen via altered fear extinction recall, fear renewal, and fear reinstatement. All that's seen in PTSD patients, but also can be replicated in animal model of PTSD. What I have not talked about today, but there is an emerging evidence that contextual processing abnormalities are actually present in PTSD patients in other functions that are not limited to fear learning. These deficits in contextual processing could contribute to the maintenance of fear memories, other intrusive, avoidant, and hyperarousal symptoms that I discussed, but also abnormal fear learning of novel stimuli, hypervigilance, and re-traumatization. Thus, constitute a more general and more parsimonious model that doesn't require multiple systems and multiple pathophysiological processes. Furthermore, animal work implicates glucocorticoid, glutamatergic and glutamatergic signaling in hippocampus and medial prefrontal cortex in these deficits, as well as excessive noradrenergic signaling from LC to hippocampus during REM sleep. This really provides a platform to integrate pre-existing biological findings and that already have been reported in many studies in PTSD with central CNS model and try to understand it together. In addition to implicated novel function in circuitry, it also implicates cellular and molecular processes, as I mentioned, as potential target for therapeutic and preventive strategies. Now that allows many different approaches to that, because many different pathways could lead to this hippocampal-thalamic-prefrontal dysfunction that leads to context processing. It could be genetic or genomic contributions. There could be deficit in neurogenesis in hippocampus. There could be sleep deficits that primary to LC. We could have additional processes, even additional processes within amygdala that produce, for example, diminished extinction that also can lead to abnormal contextual processing. So this model, as I mentioned, offers new targets for study and for development, and that raises additional hopes for developing novel interventions and novel preventions that is urgently required. As I mentioned before, this work has been performed by many collaborators. My collaborators in functional neuroimaging laboratory, Steve Taylor, Robert Welch, Chandra Sripada, Tony King, and others. Our animal laboratory with Chia Chen, Mala Rajaram, Johnny Shainen, Ellie Rodriguez, John Greco, and Haley Briggs, as well as graduates, those who have been in my laboratory or laboratories and had since moved to independent career, such as Luan Fan, Jen Britton, Sarah Garfinkel, Samir Khan, Diane Knox, Becca Sripada, and others. Collaborators, my main collaborators into this work are Gina Poe, Jim Abelson, Steve Marin, Arik Shalev, as well as Shane Perrine from Wayne State. And disclosures you can see before, and I do appreciate the extensive funding that we have received both from NIMH, DoD, and VA to study those topics. And thank you very much, and I'm open for questions.
Well, Dr. Lyberson, thank you so much for this highly informative and topical presentation. It looks like there's no questions this time, and as we're approaching the end of the hour, I would just like to invite people to submit questions either via email, either to Dr. Lyberson or to Cohen Veterans Bioscience on our website. And again, Dr. Lyberson, thank you so much for taking time today for this great talk. And thank you everyone for participating.
Thank you very much.